terus masuk kan?
Kita video.
Saya dah boleh start ke? Dah lah, dah masuk. Okey. Saya kedengar. Bismillahirrahmanirrahim. Alamak, macam mana nak cancelkan notification? Tapi saya share screen sebab keluarkan notification. Alamak, keluar. Sebab saya dekat sini, kita buka mic. Buka mic. Kak Ida buka mic. Dah buka. Okay, Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. Okay, ada MRSM apa hari ni? Yang join kelas kita hari ni, webinar. Okay, sebelum saya mulakan kelas pada hari ni. Lama, adakah saya punya mic di unmute kan? Okay, sebelum saya mulakan kelas pada hari ini, saya ingin ucapkan terima kasih kepada KPP kita, Puan Tanti dan juga Cik Fauzi sebab beri peluang ni untuk saya share sedikit ilmu berkenaan menjawab paper IGCSE 0610 Component 6. Okay, so introduction about me. Hi everyone, I teacher Wahida. Ada lag sikit, sorry belum keluar. Okay. Hello, I teacher Wahida. I'm from MRSM Tun Abdul Razak. Okay, I've been working with MRSM and with Mara since 2003. Okay, before that, saya pernah kerja as research assistant dekat Uniscience Holding and then become an education officer di Parinda Wetlands. Then pada 2003, I start working as a teacher, biology teacher in MRSM Langkawi. Okay, 2010 baru saya pindah ke MRSM Tun Abdul Razak sampailah sekarang. So sedikit information about me. I love hiking. I wish I can travel around the world, and for now I run for my fitness. Okay, so before kita start, always I remember this, guna bangun pagi, wake up and go to work on yourself before you go to work for anyone else. So keep your fitness great, okay? Okay guys, hari ni kita nak fokus kepada Biology 0610/63. This is alternative to practical paper. Okay, I hope that you have the knowledge already, information about what you are taking in November series nanti. Kita ada biology, ada 3 paper kan? Which is the 0610/23, paper 2. Ya, ada lag sikit kat I punya slide ni. Okay, 061043 paper 4, and today kita nak fokus kepada 0610/63. Okay, this is all about experimental question. Normally there will be 2 question for experiment ni, but sometimes Cambridge boleh keluarkan sampai 3 soalan dalam satu paper. Okay, I hope that everyone has ready with the handouts yang I give semalam. Harap cikgu-cikgu daripada maktab lain dah distribute the paper to everyone dekat maktab. Okay, ada 2 set of soalan dekat dalam handouts yang I share semalam dan 2 tambahan daripada previous year's question. Okay, time allocated untuk jawab paper 6 ni adalah 60 minit. Okay, total mark is 40, and this paper will give you weightage at 20% of total mark for 0610. It's mean that setiap soalan yang kita jawab betul, kita dapat 1 markah. So markah tu menyumbang sebanyak 1 markah untuk total syllabus mark. Okay. Alright, sini Cica nak ada sikit macam mana you nak tahu berapa markah yang kita kena score untuk paper 6 ni untuk menjamin kita dapat A star for biology 0610. So kat sini I have simplified the grade threshold since November 2018. Okay, as kita tahu kita nak ambil exam in November kan. So November series kita akan compare grade threshold for November series. Okay, kat sini compilation 2018. As you can see from my screen, November 2018 to get A for biology 0610, you must score 33 marks out of 40 marks. Kita tahu tadi total markah paper 6 adalah 40 kan. So to get A, you kena score 33 markah. 2019, the grade threshold naik sikit, which is you can dapat 34 out of 40 baru dapat A. But since COVID ni, dah ada penurunan pada grade threshold, see 32. Now last year, November 2021, grade threshold to get A only 30 marks out of 40. But takkanlah kita nak skor 30 je, sedangkan kita boleh skor 40 markah. Kan? So try your best. Jangan target rendah sangat 30 ni. Manalah tahu tiba-tiba naik balik kita punya grade threshold kan. So always aim for the highest. Kalau boleh 40. Tak susah pun nak jawab paper 6 ni, senang je. Okay. So this is our cover of our paper. This is paper 0610/62 May June 2021. I ambil contoh je. Okay, what is 0610? That is the syllabus code. Okay. Number 6 over there. Refer to paper apa kita nak ambil. This is paper 6. And number 2 at the back is the variant. Okay, as we understand, Cambridge always produce 3 variant of question paper, variant 1, variant 2, variant 3. For June series, they will take variant 2. Okay, for November series, that is our paper coming soon, is variant 3. Okay, tak ada banyak beza pun antara variant, bukan tak ada banyak. Ada perbezaan between this question, tapi kita tak boleh pilih variant mana yang kita nak ambil. This one will be ditetapkan oleh Cambridge. Okay, this is a very important advice to you whenever you nak jawab paper 6. Please be prepared with black, use black or dark blue pen. Okay, jawapan use black or dark blue pen. Okay, and then whenever you want to draw graph or draw diagram, you must use HB pencil. Okay, for drawing graph and picture yang dia minta kita lukis. Okay, dalam paper 2 also use HB pencil when you want to shape the question yang jawapan yang you nak transfer ke dalam the OMR paper tu. Okay, use HB pencil because HB pencil bila kita padam dia tak smudge so that dia tak ganggu kita punya diagram yang sebenar. Okay guys, awesome. Paper 6, fokus kepada awesome paper 6. Yesterday kita dah fokus pada paper 4, today fokus pada paper 6 dan kita aim at the end of this session hopefully everyone will score 40 marks. For our coming mock ni kan 29 August nanti, I hope that everyone can score 40 marks. You have plenty of time, not really, tapi boleh lah kan kita try to use whatever we learn today to score 40 marks for paper 6. Okay, as we know, bila kita nak understand macam mana cara nak jawab, kita kena familiar. Okay, familiarize with the common words. Bila kita dah tahu faham apa common words tu nak, baru kita boleh bagi jawapan yang tepat kan. So untuk paper 6 today, Cik akan share with you how to answer common word planning experiment. Ni yang paling ramai orang takut kan. Padahal tak susah pun, senang je. Planning experiment ni nak score 6 marks. Easy peasy. Okay, tabulating data. Ya Allah, yang ni paling senang. Okay, 3 ke 4 markah. Banyak pula tu markah dia. Kalau kita score this one dah dapat 10 dah. 10 dalam tangan guys. Okay, conclusion pun tak susah. 1 markah je. Patutlah dia bagi 1 markah sebab soalan ni memang tak susah sangat pun. Okay, and then ada sedikit calculation. Calculation sometimes will give you 3 to 6 marks of the question paper. And then drawing. Yang ni memang soalan wajib keluar, mesti akan ada drawing. Drawing in biology tak sama macam you draw yang artis-artis tu kan. Apa tu nama dia? Seni visual, tak sama. Okay, so drawing always 3 to 4, sometimes 5 marks. It can be until 5 marks bila dia minta kita label the diagram. And then how to draw a graph. Yang ni senang je insyaAllah. Okay, 4 to 5 marks. Okay, this is what we are going to cover today. Hopefully sempat lah. Alright. First thing first. Sekarang je dah masuk dalam dewan exam. Okay, kita dapat paper. First thing first, when you dapat paper, jangan gelabah. Cool down, relax. Memang kita ada 60 minit dan kita akan score 40 marks ni. First, apa kita nak buat? Read the question carefully. Okay, carefully, tenang-tenang, jangan kalut sangat. And then while you reading the question paper, underline and identify the MV, RV and the CV. Dalam soalan tersebut, cari, underline and then labelkan. If there is, sometimes dia bagi kita source of error dekat dalam steps yang dia tunjuk dekat dalam question tu. So kita identify siap-siap, label siap-siap. Alright. For example, now everyone please refer to the handouts given. Turn to page 2. In page 2, you will jumpa this question. Boleh tak? Okay, I give you some time to cari this. Jumpa kan? Alright. Cikgu-cikgu kat maktab semua dapatkan? Boleh bagi semua dah dapatkan question ni? Alright guys, boleh kita baca sama-sama? Refer to the handouts given, okay? Turn to page 2. Alright. In this question, we read it together, okay? A student investigated the effect of surface area on the rate of diffusion. Do you find something in this phrase, in this ayat? Ada tak? Kita jumpa apa? So we can label the effect of surface area. This is the MV. On the rate of diffusion, so rate of diffusion is the RV. So sambil baca, kita underline dan kita labelkan. So that later, if there is question asking you about manipulated variable, tak payah nak susah pergi balik dan jumpa dah. Salin je balik ayat tersebut dalam space yang diberi untuk jawapan, kan? Okay. Alright, now kita dah ada MV, kita dah ada RV. The student prepare different size block of agar. Different size of block of agar ni pun adalah MV. Alright. The block of agar had different surface area. This is also MV. The agar contained universal indicator which made the agar green in color. So dah ada color. The block of agar were put in hydrochloric acid. Universal indicator turned red in the presence of acid. As the acid diffused into the agar block, the indicator changed from green to red in color. Oh, kita nak observe the changes in color from green to red. Alright, next. The time taken for the acid to diffuse to the center of the block was measured. Siapa tahu what is this? Ada ke attendees yang pegang phone yang boleh jawab kat teacher sekarang? Okay. The time taken for the acid to diffuse to the center of the block was measured. This is what? Ni kalau depan-depan, I boleh bagi hadiah kan? Bagi coklat. Siapa dapat jawab? Okay, the answer is, answer lah. This is what? This is RV. Okay, time taken. Time taken tu adalah RV. This is what we measure at the end of this experiment. Okay. Alright, next. Okay. 10 centimeter cube of 1 mole per decimeter cube hydrochloric acid was put into each of 4 test tubes. Dia nyatakan 10 centimeter cube. This is the concentration of hydrochloric acid. Dia nyatakan 10 dan dia letak put into each test tube. This will be our constant variable ataupun fixed variable. Alright. The student use a ruler and a knife to measure and cut 4 blocks of agar A, B, C and D from a larger piece of agar, and then each block was a different size as shown in figure 1.1. Ni dia punya information sahaja, and we know that the agar has different sizes. Okay, A, B, C, D referring to different sizes of agar. Alright. Okay. Sambungan dia in figure 1.1. Step 3. Agar block A was placed into one of the test tube containing hydrochloric acid. A stop clock was started. Okay, this is the beginning of the RV kita kan. The color of agar block A was observed. Once they change color from what to what? Siapa boleh jawab? Nak observe color berubah from what to what? From green to red. Kan? Okay. The time taken for agar block A to change in color from green to completely red was recorded. Again, this is our RV. Step 3 was repeated with the remaining block B, C and D. This is the manipulated variable. Kita sambung lagi. Alright. So, figure 1.2 shows the time taken. Dan kita dah banyak line kan? Korang dah banyak ke? Laju sangat ke teacher bagi tahu ni? Boleh tak korang underline the question? Boleh ya? Alright, figure 1.2 shows the time taken for each agar block to become completely red. And then, okay, come to the question. Kat sini ada berapa command words. Dia suruh kita buat apa? The first, they ask us to prepare a table. This is the first command words. Prepare table and record the result. This is tabulating data. Thank you Adik Najmi from MRSM Batu Pahat. Hi MRSM Batu Pahat. Okay. So first question dia suruh kita prepare table to record the result. Right. And then dia suruh buat apa? Convert the time shown in figure 1.2 to second and record them in your table. Okay, tukar kepada second and then record them in your table. Ni yang satu tadi lah. Satu dia suruh kita prepare table. Lepas tu kita suruh kita record. Lepas tu dia suruh kita convert. Okay, from minute and second tu all tukar kepada second. So dapat this is our question. So first command word kita nak cover today is the tabulating data. Macam mana nak buat table? Senang aja guys. Okay, in tabulating data, nak buat table, mesti ada column kan? So the first column, always kita letak MV, the manipulated variable ataupun independent variable. Independent variable sometimes ada unit, sometimes tak ada unit. Contoh macam agar block tadi, just agar block kan. Kalau kita nak label agar block tak ada unit. So MV sahaja, nyatakan sahaja MV dia without units. Okay, the second column always is RV plus unit. So RV kita always ada unit. So jangan lupa letak unit. Wajib, baru you boleh dapat markah. Alright. Bila kita record the data, data must be uniform. If your data has one decimal point, so all data inside the same column kena ada decimal point yang sama. Okay, barulah dia panggil uniform data. Yang ni wajib. Kalau tak uniform data, dia terus hilang markah untuk satu record the data inside the table. So rugilah. Alang-alang dah supaya buat table kan. Jangan tak uniformkan data pula. Alright. When we draw table, mesti kena guna ruler. Okay, mesti kena guna ruler and also pencil. Kenapa kena pencil? So that if there is extra line, kita boleh padam. Alright. This one from the examiner itself, daripada examiners yang bagi training kepada semua cikgu MRSM, dah state kalau ada extra line dan dibiarkan sahaja, memang kena tolak markah untuk table MP1. Marking point number 1 terus tak dapat lagi kan. So jangan biarlah. Okay, we take this notes. Okay, this is an example of a table. The first mestilah MV. Ada unit in bracket. Kenapa unit in bracket? Sometimes ada unit, sometimes tak ada. So kalau tak ada, tak payah letak unit dia. Alright, the second ORV, operational responding variable. Apa yang kita buat? Okay, kalau nak buat dua lah. ARV is apa ARV? Okay, responding variable yang kita kena abstract, yang kita kena calculate menggunakan formula. Okay, kalau kita letak dekat last column, baru kita letakkan ARV. Yang ni, apa yang you kena calculate, letak kat sini. Okay, the third column. Okay, ni basic. Bila kita nak tabulate data. First column MV, kalau ada dua je, second column RV, settle. 5 markah, 4 markah dalam tangan. So, senang kan? Alright. So, kita test, tengok. Check balik kepada page 3 in your handouts. Okay, page 3 in your handouts. Cuba buat soalan ni sekarang. Boleh dapat tak sama macam yang Cik nak tunjuk lepas ni? Okay, everyone take your time. Cuba buat table dan skor markah untuk this question. I hope that everyone ada paper sekarang. Boleh buat tak? Okay, cikgu-cikgu dekat MRSM, dekat MRSM Batu Pahat. MRSM mana lagi ada ni? Cuba say hi dekat Q&A tu. Saya nak tengok MRSM mana yang hadir ni. Sambil-sambil you buat the table. Nanti I share the marking scheme. Ada tak? Tak ada lagi. Okay. Alright. This is the answer for this question. Okay. The first agar block. Mana kita nak dapat agar block ni? Kat sinilah. Okay. Time taken for each agar block to become completely red. So you ambil this one. Time taken to completely red ni, masukkan dia dekat dalam RV. Sebab ni memang RV kan? Okay, agar block kita ada dalam gambar tadi, agar block A, B, C, D kan? So kita transfer agar block tu dekat sini. So this is the MV kan? Different size of agar block. Okay, so dia duduk kat paling kiri. The second one is the RV. Okay, unit kita kat sini adalah second. So second macam ni cara tulis dia. So teacher dah buatkan dia punya result macam ni. Semua orang dapat sama dengan I ke? Kalau sama, Alhamdulillah. Okay. I show you some of my student punya kerja. Diorang buat dah soalan ni dan ini hasil dia. Sebenarnya ni adalah hasil kerja daripada MRSM KKB last week. Okay, I have discussion with them, seminar with them. So, teacher pinjam tau kalau ada MRSM KKB kat sini. Saya pun punya agaknya ni. Okay guys. So, berapa markah for this student? Alright. MP1, marking point number 1. Okay, this is the marking point number 1. Table drawn with a minimum of 2 column and a header 4 line. And then 4 line. Okay. Ada tak dia 2 column? Yes, ada 1, ada 2. A header and a header and then 4 line. 1, 2, 3, 4 lengkap. Okay. Tapi masalah dekat sini untuk dapatkan MP1 dia kena ada table drawn. But for this answer tak lengkap kan dia punya table. Okay. Tak sempat tulis gariskan this one kot. Okay. So because of tak ada yang ni, this answer tak boleh being rewarded with MP1. Okay, no MP1 for this one. So MP1, marking point number 1, kosong. Okay, second marking point, appropriate heading with unit. Okay, but okay tak dia punya heading? Surface area. Surface area ABCD. ABCD apa sebenarnya? Dalam question, ABCD ni adalah agar block with different sizes kan. Tapi dalam kita punya diagram sebelum ni, yang kita tengok sebelum ni, ABCD ni adalah the agar block. So surface area ni tak sesuai. Kalau surface area kena ada unit, betul tak? So this one dah tak boleh dapat markah untuk appropriate heading. Okay, rate of diffusion. Tak ada pun question to ask for rate of diffusion. Dia cuma minta kita record the time yang telah kita dapat tu. So tak bolehlah bagi rate. Rate ada calculation dia sendiri. Yang ni lah ARV. Tapi dalam eksperimen ni kita tak disuruh untuk kira ARV. Hanya ORV saja apa yang kita nampak. So and then unit. Unit ni betul. Tapi because of heading ni dah salah. Jadi MP2 pun tak dapat. Okay. Recording of 4 reading time. Okay, ada tak reading time dia? Yes, ada. 1, 2, 3, 4. So, dapatlah MP3 kat sini. Alright. MP4, correct conversion to second. So, tadi I dah kira. So, sama macam I punya ni kan. So, dia dapat kat sini. Semua sekali betul for MP4. This is how nak marking dan nak score marks. Okay, so this student is awarded with 2 marks only. Okay, Okay, MP3 dapat 1. Okay, MP4 dapat 1. So total 2 marks out of 40. Betul tak? Soalannya ada 4 marks. Kita ingat berapa markah dia, tapi I rasa betul 4 marks markah dia total. Okay guys, how about now? Okay, semua orang sempat buat tak? Kita tengok satu lagi contoh, okay? So, this one. Dapat tak this student MP1? Dia ada block, should be dia tambah apa kat atas ni? Agar block. Rate of diffusion. Dia punya table dapat MP1 untuk table. Okay, tapi rate dekat sini dah salah. So MP2 tak dapat. Okay. Ada tak 4 recording of 4 reading? Yes, ada 1, 2, 3, 4. This is good. MP3 and all are correct. That is MP4. Okay. Alright guys. I hope that everyone clear now macam mana nak buat table. MV for the first column, second column is our RV. Jaga when we nak tulis the heading. All the wording you can get it from the question itself. Okay. Easy peasy je, senang je kan. Okay. Alright. Next, I nak terus masuk kepada conclusion sebab question ni nak jawab conclusion kita kena faham apa eksperimen kat atas kan. So alang-alang kita dah tengok eksperimen tadi, kita terus tengok command words yang kedua, that is conclusion. Okay, to answer conclusion question, okay, you must able to write what changes can you see from the experiment, and your answers mesti ada MV and also the RV, the changes that you observe. Okay, what do you think is the conclusion for the experiment yang kita baru baca tadi? Ada anyone nak jawab? I tengah tengok Q&A ni. Sunyi je Q&A, tak ada siapa-siapa tanya. Tak apa teacher, sebab ni senang. Bahagian depan ni senang kan? So, what is our conclusion for this experiment? The bigger the agar block. Boleh tak? Okay. The side length of the agar block. Okay. The time taken for the agar block to change color increases as the side length increases. So, agar block A, B, C, D tu kan dia mempunyai different sizes kan. If you say different sizes of agar block have different time, tak sesuai. Okay, untuk conclusion kita dah tahu dah. Ada yang besar, ada yang kecil, terus sebut. Okay, time taken of the agar block to change color increase as the side length increases. Okay, the bigger the agar block, the longer the time taken for the agar block change color from green to red. Boleh juga. Asalkan ada MV, ada RV. Okay? Alright. So kita tengok kepada question yang seterusnya. This is on page 14 of your handouts. Okay, eksperimen yang kedua, set of question yang kedua. Boleh? Alright. Can we read this together? As the surface area decreases, the time taken will decreases. The time taken what? The time taken for the agar to change color from green to red also decreases. Boleh? Okay, thank you Adib Najmi from MISM Batu Pahat. Okay. Boleh kita go to the next question? Boleh kita apply sekali lagi teknik yang pertama tadi, the tips yang pertama tadi. We read the question, we underline, and kita identify MV, RV dan CV. And take note juga if there is source of error. Okay, for the second question, read it together. Semua orang ada question paper kan? Okay, a student investigated the effect of temperature. Bila dia start je dia kata effect of temperature ni, kita tahu dah. Ni mesti nak jadi dia punya MV. Effect of temperature. Kesan temperature. So temperature diubah-ubah. So kita tengok kesan dia apa. Okay, effect of temperature on the rate of respiration. Kat sini baru jumpa rate of respiration in yeast cell. When yeast cell respire, they produce carbon dioxide gas. Selain daripada carbon dioxide, yeast buat apa lagi? Dia release ethanol, kan? Alright. Okay, in this investigation, the gas was collected in a balloon that was attached to a test tube containing a yeast suspension. Okay. Step 1. The student prepared a hot water bath and a cold water bath. This is what? Handling the MV. So this is MV. Ada hot, ada cold water bath. The student measure the temperature of the water in the hot water bath and in cold water bath. Measure guna apa? Thermometer. A yeast suspension was stirred with a glass rod. 25 centimeter cube of yeast suspension was put into each of 2 test tube. Okay, ada 25 kat sini. This going to be our CD. Okay, dinyatakan nilai tu. And then dia kata each test tube letak 25 centimeter cube. So that is going to be constant variable. Okay. A balloon was stretched over the top of the test tube. One test tube and balloon was put into a hot water bath and other test tube and balloon was put into the cold water bath as shown in figure 1.1. Tengok gambar seterusnya. Macam ni rupanya bentuk eksperimen dia. Okay, step 6. The circumference of each balloon was measured by placing a piece of string around the widest part of the balloon as shown in figure 1.2. A ruler was then used to measure the length of the string that wrapped around each balloon once. Okay, this is what? What is this? What is this? Thank you, Taufik from MRSM Tun Mustafa at TMT. Okay. This is what? This is R D. Okay. Okay guys, next question. And next, sambungan kita punya bacaan ni. Step 7. The circumference of each balloon was measured at 0 minute, 5 minute, 10 minutes and 15 minutes. The student result are shown in figure 1.3. Dia dah bagi dah result kita macam ni. And then dia tanya kita, boleh tak? Boleh tak? Dia suruh kita buat apa? First, prepare table. Second, record the result shown in figure 1.3. So guys, dia dah cakap shown in figure 1.3. Focus only towards figure 1.3. So, kita nak lukis yang ni je. Kita nak convert, kita nak transfer data kat dalam figure 1.3 ke dalam table kita. Okay, boleh tak kita buat table ni? Bear in mind, our MV adalah temperature. Temperature kita apa? Hot 1, cold 1. Tak ada nilai pun, just hot and cold. So that is our MV. Kita ukur selang minit. Okay, dan kita ukur apa? Circumference. Unit dia apa? mm. So, can you draw your table now? Can you do this question now? Cepat, siapa dulu? Teacher show jawapan dulu ke korang sempat buat dulu? Do it now. Try. Come on. Ada ke cikgu boleh ambil gambar hasil pelajar masing-masing? And then boleh share dekat Telegram ke, dekat WhatsApp group ke? Online pun kita boleh interact kan? Anyone nak share? Wah, I nampak MRSM Kuching. Hai MRSM Kuching. Ada ke pelajar daripada MRSM Kuching nak try jawab ni? Boleh ke cikgu Aliti share jawapan student? Boleh send dekat group, WhatsApp group. Tapi kan lambat kan, nanti tak sempat kita nak habiskan. Banyak lagi command word kita. So teacher show, this should be our answer. Sama tak? Sama tak? Sama teacher. Thank you. Okay, next. Okay, this is an example of question, jawapan pelajar yang saya pernah terima untuk this question. Okay. So, the marking scheme state that, okay, table drawn with a line between heading and data and at least ada 3 columns. Okay, ada tak 3 columns? Ada 1, 2, 3. So, markup untuk MP1 dapat tak? Yes, dapat. Pandai. So, dapat markup untuk MP1. That is drawing the table. Okay, second marking point. Column or row heading with unit. Unit dia apa? The first one is time. The unit is minutes. The second is circumference of or length. And unit dia mm. The third one is hot and cold. That is what? That is our MV. MV kita dia letak yang last. Patutnya dia yang letak yang pertama sekali kan? Okay, boleh je. Okay, kalau dalam jawapan teacher tadi, korang nampak tak? Kat mana MV dia? MV dia dekat sini. So you can put it here or you boleh je letak kat awal kat sini. Boleh je mana-mana. But is this the simplest one, I think? Nanti I'll show you macam mana kalau kita nak letak coal tu dekat sebelah paling kiri sekali. Okay. Next. Berapa kawan yang dapat ni? Time. Yes, betul. But cara dia tulis unit tu tak betul. So dah salah kat sini. And then cold. Betul. Tapi cold takkanlah unit dia mm. Okay. Kat sini your friends lupa nak tulis. This is what? This is the circumference of the balloon. Unit dia mm. So kat sini dah tak cukup information yang diletak on the header. So tak boleh nak dapat MP2 because MP2 dia dah nyatakan kena ada heading like this together with the unit. Alright. And then number 3, MP3. 8 circumference recorded correctly and time recorded correctly. Okay, ada 8 circumference. 1, 2, 3, 4, 5, 6, 7, 8. Dapat MP 3. Okay, time recorded correctly. 1, 2, 3, 4. Yes, barulah dapat MP3. Okay, kenapa saya gunakan jawapan ni untuk show to for you? Because dalam IGCSE, you cannot write. Ni satu kesalahan besar tau. You cannot write unit in the data. Units only need to be written in here on the heading. Inside the heading sahaja. Okay, yang ni semua tak boleh. So I risau if you do like this, you might lose your MP3. Hilang markah MP3, sayang. Bukan tak betul ni, tak betul dah. Tapi because dia letak all this unit ni tak boleh. Tak boleh. Memang salah besar kalau kita letak unit tu dalam kita punya data. This one also tak perlu. Okay, boleh tak? Everyone clear? Kucing okay? Emerson kucing okay? Boleh ya? Okay, can we proceed? Another. Okay, I believe some of you mungkin cuba untuk jawab like this. Tak salah pun, betul. Okay, this one. Betul kan this one is the MV? Yes, betul MV. Tapi dalam MV kita tak ada unit kan sebab dia just cakap cold and hot. So tak perlu letak degree Celsius. Okay, you go. Tapi dia tak letak nilai. Tapi temperature ada unit. Nak selamat letak degree Celsius. Boleh. Walaupun dia tak bagi kita nilai. But I believe tak apa je in this case because dia bagi kita cold and hot. Alright. Next is the time taken. Time taken ni betul. Okay. First I bagi dah MP1 sebab ada table yang nice. Bila nak letak MP2, time taken tu betul tapi cara tulis unit ni salah. Macam mana cara tulis minit? Kena tulis minutes. Contoh. Circumference of what? Circumference of the balloon. Tapi circumference of the balloon dia bagi unit yang betul, dia betul. But sebab ada satu yang salah, so tak dapatlah MP2. Rugi kan? Alright. And then ada tak untuk data for minutes? Yes, ada 1, 2, 3, 4 lengkap. And then ada 1, 2, 3, 4, 5, 6, 7, 8. So this one will get MP3. Okay. MP3 dapat, MP2 dapat 0. So total mark is 2. Betul tak question ni ada 3 markah? I hope that's what saya dah lupa berapa total marks dia. So boleh dapat? Okay, siapa yang cuba buat macam ni pun betul, tak ada masalah. Okay, sebab tadi kita belajar the first one is the MD and then the RV. Kan? In this case, kita ada another one that is the time. Alright. Everyone okay? So conclusion for this experiment. Okay, bila nak tulis conclusion, kena ada MD, kena ada RV. Siapa nak try tulis? Adik tak nak try tulis ke? Type lah jawapan you. What is the conclusion for this experiment, guys? Thumbs up. So this is the answer. So in hot condition, so kita ada dua je kan, hot dengan cold kan. So in hot condition, yeast cell respire more ataupun yeast cell produce more carbon dioxide. Okay, yeast cell produce a higher volume of gas ataupun higher volume of carbon dioxide. Kena ada perkataan produce. Okay, yeast cell respire and produce more carbon dioxide. Boleh. Ada MD, ada RV, dapat markah. Clear? Okay. Dah 3.43 dah. 43 minit dah. Okay, baru sampai kat sini. Next. Yang ni yang paling best. Best ke? Best sangat. Planning experiment. Tak susah pun bila kita tahu cara jawab dia. Okay, so in planning experiment, what you must consider when you read a question, bila you baca question, you ask yourself, in this experiment, given experiment, what is being changed? What is being changed to apa? MV, right? Second one, what is being measured or how to measure? And then, what is being kept the same? Constant, CV. Okay, and then how to do this experiment, method to handle the experiment. And then how many times the experiment should be repeated. Nah, berapa kali nak ulang supaya kita dapat result yang reliable. Okay. And what is, last one, what is the suitable safety precaution during handling the experiment? So ada berapa point dekat sini yang kita kena ask ourselves bila kita nak plan experiment. Ada 1, 2, 3, 4, 5 and 6. 6 marks. As easy as that. Okay, so let's go. Kita baca dulu question ni. Daripada question, kita boleh identify the MV and also the RV. And then kita boleh plan what is the CV. Okay. Photographic film consists of a plastic sheet coated in crystal. The crystal are fixed to the plastic sheet by gelatin, which is made of protein. If the gelatin is digested by protease enzyme, the crystal fall off and the film will become transparent as shown in figure 1.4. This is the before protease treatment. Nampak tak semua hitam? And then after certain time, the film become transparent. Okay. Boleh kita buat eksperimen ni? Boleh. So next question dia kata, plan an investigation to determine the effect of temperature on the activity of a protease enzyme using photographic film. So, dah nampak ke? What is the manipulated variable here? Daripada soalan ni. Bila you start jumpa question, dia begin with plan and investigation. Itu adalah the starting point kita nak cari kat mana MV dan RV yang kita boleh guna pakai. Okay, dalam case ni, the effect of temperature is our MV. on the activity of protease. Activity of protease adalah kita punya RB. Okay, do you get the idea? Macam mana cara nak jawab ni? Okay, the first question yang berliga dalam kepala kita macam yang I stated tadi, what is being changed dalam eksperimen ni? Dia kata effect of temperature. So temperature lah kita nak ubah-ubah kan? Okay, what is being measured? Activity of protease. Macam mana nak measure activity of protease? Berapa lama ya dia ambil protease ni, ambil untuk make the film from warna black until transparent. So kita tak boleh ukur time, kan? Right. Okay, terus show the first, kita dah underline kan, so kita terus tengok the first step. Bila kita baca, kita nak identify what is being changed. This is considered as independent variable. Okay, dalam IGCSE, when you want to plan an experiment, independent variable ni, you must give at least 2 or 3 value plus unit. For example, dalam question ni, dia minta kita temperature. So, you kena bagi at least 2 or 3 value of temperature. Okay, dalam eksperimen ni, macam mana kita nak tulis the first part for us to get markah for manipulated variable or independent variable. Macam mana nak tulis? Macam mana kita nak tulis MV dia? First nak start. Temperature apa kita nak letak? Kita ada idea tadi, dia bagi kita protease. And that protease boleh digest the protein kan? Barulah bila dia digest protein, nanti film tu jadi transparent kan? So, bila ada enzyme, kita tahu enzyme sensitive towards temperature. Kita nak pilih berapa temperature? 2 ke 3? Kita letak 3 lah. So, enzyme, activity enzyme, dia sangat terkesan dengan temperature. The first, yang paling bagus, enzyme work best in what temperature? 37 degrees Celsius kan? Why not kita letak 30 degrees Celsius? Enzyme dia tak aktif kalau 10 degrees Celsius. Enzyme akan denatured bila 60 degrees Celsius. Boleh kita choose macam ni? Ada 3 tak data kita sekarang? Yes, ada 3. 1, 2, 3. So, how to write sentence nak refer kepada independent variable ni? I bagi contoh. For example, like this. Okay. Prepare 3 water bath with different temperature which is 10 degrees Celsius, 30 degrees Celsius and 60 degrees Celsius. You boleh tulis this one, you terus boleh dapat 1 marks untuk MV, manipulated variable. Dapat tak everyone? Macam mana cara nak tulis the MV? Just state, prepare 3 water bath with different temperature which is 10 degrees Celsius, 30 degrees Celsius, 60 degrees Celsius. Tak perlu you nak explain, ambil beaker, letak water, letak ice to make the temperature 10 degrees Celsius. No need. Tak payah explain that one. Just cakap je prepare. Dia suruh plan, bukan suruh buat. Okay. So plan je. So prepare 3 water bath. Boleh? Okay. Idea, tips. Whenever question to ask about temperature, terus state kat situ how you make sure that the temperature tu boleh stay at 10 degrees Celsius. So cara kita nak handle supaya dia tetap pada suhu yang kita nak is we insulate each water bath with cloth. Okay, insulate with cloth. So that when we set the water bath at 10 degrees Celsius, using insulation, so dia akan maintain at 10 degrees Celsius, tak berubah. Okay? Alright. Next, what is being kept the same dalam eksperimen kita ni? Okay, this is called as fixed variable ataupun constant variable. Dalam constant variable, bila nak tulis planning eksperimen, nak sebut pasal fixed variable ni, gunalah perkataan same. Okay, same. Okay, use word same volume or same source or same mass at the beginning of the experiment. Okay, kalau ada unit, better sebut unit dia. Okay, dalam kes kita ni, cut film into 1 cm each. Kan kita ada film tu kan, kita nak pakai banyak pieces kan, sebab kita ada 3 temperature. So, potong setiap satu tu 1 cm. Senang je kan? Cut film into 1 cm each. Terus dapat satu markah untuk CV, constant variable. 10 cm cube protease is poured into test tube A ataupun into test tube. Dah ada 10 cm cube. Okay, dah dapat satu lagi markah untuk CV. Biasanya CV ni dia bagi 2 markah. Sometimes 1, sometimes 2. Okay, can be CV like this. Alright. Boleh? Tadi dah cari MV, now dah cari CV. Next kita nak, macam mana kita nak buat eksperimen tu? How to do the experiment? Okay, this is called as method. Dalam marking scheme, dia akan state this a method. Okay, handling apparatus and material. Macam mana kita nak handle? So, in our case today, example of this, alright, immerse the test tube A into water bath at 20 degrees Celsius. Okay, immerse the test tube A into water bath at 20 degrees Celsius. Betul kan? Ni kita handle kan? Tadi kita dah tuang dia, now kita ambil test tube tu, we put into the water bath. Set aside for 5 minutes. Set aside for 5 minutes ni pun dah jadi CV dah sebenarnya. Okay, tapi this one will be method. Okay, then put 1 cm after 5 minutes. Why we put this test tube A into water bath 20 degrees Celsius for 5 minutes? Siapa tahu why? Why we immerse this into 20 degrees Celsius for 5 minutes? Apa kita punya MV tadi? Different temperature kan? So kita nak tengok how this protease work in different temperature. So sebab kita nak detect dia kerja in 20 degrees Celsius. 20 kita tadi, 10 tak? Okay, tukar balik 10 degrees Celsius kan? Kalau kita nak tahu dia kerja dalam 10 degrees Celsius, so we need to make sure that the protease now, 5 minutes ni, the protease will have same temperature with the water bath. Okay, the protease ni akan sama dengan temperature dekat luar dia, kat dalam water bath tu. Kalau water bath adalah 20 degrees Celsius, so after 5 minutes, we hope that the protease inside here also will have 20 degrees Celsius juga. Okay, that's why we set it aside for 5 minutes. Alright. Then put 1 centimeter long film into test tube A. Once kita letak, start the stopwatch. Pendek-pendek je ayat dia nak tulis tu. Okay. Once we start the stopwatch, now kita boleh measure dia punya masa untuk the film turn to transparent. Okay. Next, what is being measured? Dah sebut dah tadi, what is being measured is the time taken. So this is RV, dependent variable. So in dependent variable, kita kena tulis how to measure and record RV using correct apparatus. Kenapa kena guna correct apparatus? Supaya kita dapat accurate result. Okay? Okay, untuk our case, we stop the stopwatch once film become transparent. So kita nak ukur perubahan color tu, kita guna stopwatch. So pada stopwatch ada nilai minit dia, ada nilai second dia. So very accurate one. So we use stopwatch. Boleh tak guna handphones? Boleh, kalau handphones tu ada stopwatch. Asalkan ada stopwatch, dah boleh dapat data yang accurate. Okay. Record the time taken for film to become transparent. Okay. This is how you tulis untuk dapat markah for RV. So far okay tak everyone? Boleh tak? Sempat tak salin dan tulis jawapan ni? Tak apa, it's okay. Boleh dia tengok refer pada skema jawapan. Cuma dalam skema jawapan dia tak tulislah cara macam mana kita menulis. Dia cuma bagi guideline sahaja. So yang teacher show dekat dalam kotak warna purple ni, is how kita nak tulis jawapan tu. Okay. Ada sangat ke? Sempat kan? Next. How many times the experiment should be repeat? Berapa kali kita nak kena repeat the experiment? Kalau kita buat sekali, do we confirm that the data is betul? Not sure juga. Cuba buat 2 kali. Let's say dalam eksperimen ni satu kita dapat 10 radius. Katalah kita dapat rate dia 5 minit je. 5 minit dah hilang dah. Lepas tu kali kedua kita buat 15 minit baru hilang untuk 20 degree Celsius. And then kita tak tahu 5 minit ke 15 minit yang betul. Kita repeat sekali lagi kali ketiga. Untuk kali ketiga we got 17 minutes. Which one you rasa yang betul? The first one ke, the second one ke, the third one? Bila kita ada 3 data, kita dah nampak the first one 50. The first one, let's say the first one is 5. Ni yang reading pertama. Reading kedua kita dapat 15. Reading ketiga kita dapat 17. Kalau macam ni yang mana satu yang betul? Sebab 5, 15, 17 ni lain-lain kan. Kalau 5 ni jauh sangat dia punya bacaan dengan the second reading and third reading. In this case seems like reading number 2 dengan reading number 3 is better. Dia sebab range dia dekat-dekat. So kalau I percaya this one dengan this one adalah yang tepat. And number 5, yang 5 ni 5 minit je ni macam tak berapa tepat. So this can be anomalous. Okay. So bila kita buat satu-satu eksperimen tu, nak dapatkan data, kita try buat sampai 3 kali, kita boleh cancel anomalous, kita akan dapat reliable data. Okay. Okay, dan dalam IGCSE, if you stick dalam planning experiment tu, you cakap repeat experiment 2 or 3 times to get reliable data and cancel the anomalous, terus dapat 1 markah untuk repeat. Satu markah tau, tak tulis apa-apa, tiba-tiba tulis repeat the experiment 3 times to get reliable data. Tu dapat 1 markah. Okay, in our case for question yang tadi tu, okay, repeat the experiment 3 times to get reliable data. Sama je data I. Macam tadi kan. So, repeat the experiment with water bath at 30 degree Celsius and 60 degree Celsius ni tak sama macam yang kat atas tau. Yang ni kita repeat untuk buat MV. I tulis ni supaya kita nampak this one to show the MV berubah iaitu 30 degree Celsius and 60 degree Celsius. If this one, baru you dapat markah untuk repeat. Untuk repeat the experiment to get reliable data. Okay, boleh nampak tak beza antara bullet 1 dengan bullet 2? Bullet this one dengan bullet ni. Boleh nampak beza ya? Okay, yang nak dapat 1 markah untuk repeat adalah this one. Okay, yang bawah ni hanyalah untuk kita handle the manipulated variable. Okay? Right, next. What is suitable safety precaution for this experiment? Kita guna temperature as a manipulated variable. Kalau guna temperature, nak buat 60 degree Celsius water bath, biasanya kita kena guna glove. Okay. So, dalam eksperimen kita kena state the precaution. Okay, untuk eksperimen tersebut. Precaution ni kita gunalah. Biasanya markah ada untuk goggle, penggunaan goggle. Kita pakai goggle ataupun kita pakai glove. Okay. Kalau saya sebut pakai lab coat, boleh tak teacher? Dalam lab kan kena pakai lab coat. Ya ke dalam lab pakai lab coat? Kena pakai dan rosak baju kita kalau kena asid kan. Tapi lab coat adalah perkara yang wajib. You masuk lab, you kena pakai lab coat when you do experiment. Kena pakai lab coat. So lab coat tidak dijadikan sebagai jawapan untuk safety precaution sebab dia adalah wajib. Okay, untuk dapat markah safety precaution, you boleh bagi other than lab coat. Pakailah goggle yang sesuai lah, pakailah glove. Sebab dalam eksperimen ni kita guna hot water. So, we can use glove as safety precaution. Macam mana cara nak tulis? Macam ni. Wear glove while handling hot water. Terus dapat 1 markah. So berapa dah markah kita dapat? Lebih daripada 6 dah markah kita hari ni kan. So this is the marking scheme for the question regarding the film tadi tu. Okay, ada 2 markah untuk manipulated variable. MP1 dengan MP2. Dia kata at least 2 different temperature. Ada tak kita sebut 2 different temperature tadi? Yes, ada. We stick 10, 30 and 60 degrees Celsius. Automatically kita boleh tanda right, dapat MP1. Ada tak kita cakap tadi insulate the water bath with cloth? Yes, ada. So dapatlah MP2. Kan? Senang kan? Allah senangnya. Tak sabar nak jawab IG ni. Okay, next. Responding variable. Ada tak kita measure time? The state. Measure the time taken to film for the film to become transparent by using stopwatch. Dapat dah MP3. Okay. Method. Ni handling tadi tu. Okay. Use a buffer. Tak ada, kita tak tulis. Equilibrate to correct temperature. Yang ni kita dapat markah untuk yang kita kata put the test tube into water bath at 20 degrees Celsius. Set it aside for 5 minutes. You get that for MP5. Okay. Ada tak kita cakap shaking? Tak ada. It's okay. Kita dah dapat MP1, MP2, MP3, dah dapat MP5. Dah ada 4 markah guys. Okay, next. Ada tak kita sebut volume of protease kita pakai? Ada. So we get MP7. Type of protease tak ada. Ada tak kita sebut size of the film pieces? Ada. Kita potong dia berapa panjang? 1 cm. So kita dapat MP8. Okay. Dah cukup dah 6 markah. Tak nak jawab yang lain pun tak apa. Jangan. Tulis sampai habis. Okay. MP9 nampak tak dia cakap 2 or more replicates or repeat investigation twice. Ni yang cakap tadi kan. Kalau dia tak buat apa-apa tiba-tiba cakap repeat the experiment 3 times. Dia tanda right, dia bagi MP9. And then MP10 is valid safety precaution. For our experiment yang ni, safety precaution will be given to wearing glove or wear goggle. Dua-dua pun kita dapat jawapan tadi kan. So dah 6 markah, maksimum lah 6, tapi kita boleh dapat 10. Kita dapat 8. Okay, this is an example of answer yang student pernah bagi. So, boleh kita marking ni sama-sama? The first ayat, jom baca sama-sama. Photographic film turn transparent after protease treatment. This is repeat the STEM question. Ada dalam STEM question, belum dapat markah lagi. Measure time taken for photographic film to turn transparent by using stopwatch. Dapat tak markah yang ni? Bolehlah bagi RV. Okay. Use 5 ml of protease enzyme. Tapi tak cakap in each test tube ataupun pour 5 ml of protease enzyme into test tube, each test tube. Kalau dia cakap each test tube, baru dapat CV. Ni macam satu je dia buat, so macam tak dapat lagi untuk constant variation. Constant variable. Okay. Varying temperature 20 degrees Celsius, 40 degrees Celsius, 60 degrees Celsius for each experiment. Since like dapat MP. Sepatutnya kena cakap lah, prepare water bath at 20, 40 and 60 degrees Celsius. Okay. Prepare 3 photographic film. Prepare photographic film tapi tak cakap berapa size. Kalau tak dah dapat dah CV. And pour 5 ml of protease enzyme on each film at 20, 40 and 60. Dia buat idea is there. Tapi tak tepat. Since like macam tak sampai. So mungkin nanti kita boleh label dia sebagai BOD, benefit of doubt. The student dia tak confirm. Padahal boleh dapat. Boleh sangat tulis. Tapi tak cukup information tu untuk memberi markah kepada dia. Repeat the experiment? Yes. This one betul. Wear glove as a safety precaution? Yes. So this answers dapat berapa markah? At least dapat 4 out of 6. Yes. 4/6 dah dapat dah. Kan? Tapi tak sesiapa kan? Tak sesiapa nak dapat. Nak dapat full mark sebenarnya. Okay, idea dah ada. Cuma kalau tajamkan sikit je terus dapat 6 markah dah. Okay, kat sini dah ada here kalau dia masukkan prepare 3 photographic film 1 cm each, dah dapat dekat sini markah. Okay, sayang. Alright guys, so next example of answers lagi sekali. Prepare 50 ml of protease in a petri dish. Okay, put 1 photographic film in each petri dish. Berapa petri dish dia ada ni? Put 1 photographic film in each petri dish. Method. Okay, leave the petri dish at temperature of 10, 30 and 60. Boleh dapat MV. Measure the time taken for the photographic film to turn transparent by using stopwatch. This is RV. Repeat the experiment 3 times. Ini dah dapat repeats. Make sure to use glove while doing this experiment. This is dapat safety precaution. Berapa markah dia dapat? 1, 2, 3, 4. 4/6. Prepare 50 ml of protease in the petri dish. BOD. Kalau I bagi BOD. Okay, tak jelas sangat. Prepare macam mana. Okay. Tak cakap nak 50 ml tu macam banyak sangat kalau kita nak letak dekat dalam petri dish. Macam tak begitu sesuai. So ada dot kat situ sama ada pelajar betul-betul jelas dengan apa yang dia nak buat. Okay. So tak dapatlah markah untuk this one, ayat yang pertama tu. Okay, but still 4 out of 6. So try your best. Nampak tak 2 markah yang ke ujung ni? Senang je nak dapat. Okay, so jangan susah hati. Planning experiment is easy. Alright, nanti I share slide ni dengan your teachers. You boleh tengok sekali lagi macam mana cara nak tulis contoh-contoh yang I show tadi. Okay. How to tulis ayat contohnya macam this one, yang color purple ni kan, you boleh rujuk this one. Ayat ni macam ayat yang selalu tulis kan. Okay guys, so kita fokus next kepada bahagian seterusnya. Ada question ke? Is everyone okay? This is not me. That is not my bag. Okay, but please focus on your goal. Do not look in any direction but ahead. Our direction is to get A star for biology IGCSE November series. Okay, not her bag. Okay. Next, kita nak tengok source of error. I hope that I have helped you to answer the planning experiment. Now kita nak move to the 4th common words, that is source of error. Lately for question sets 2021 dengan 2022, I dah tak nampak question tu directly ask source of error. Kalau sebelum ni dia memang state source of error, how to handle, how to remove the source of error. But nowadays for 2021 and 2022 dia tak tanya direct macam tu dah. Dia tukar ayat dia sikit tapi still benda yang sama. They ask for the source of error, how to overcome the source of error. Okay, can we proceed? Yeah. Okay guys. In source of error, untuk faham source of error, you can understand what caused the error. Okay. Source of error can be caused by salah pada variables yang kita set up. Okay, for example, variables yang kita pakai, the fixed variable are not fixed. Bila the variable are not fixed, then we cannot get valid data. So kita guna VAR sebagai dia punya short form dia. So variable, kalau ada kesalahan error pada V, that is the variable, so we cannot get valid data. If there is problem with A apparatus, then we cannot get accurate data. So A for apparatus to get accurate data. So kena control, kena pilih, kena choose apparatus apa kita nak pakai supaya kita dapat accurate result. So R, repeat the experiment. Why not repeat experiment? So that we can get reliable data. So VAR, senang nak ingat. V, variable, valid data. A, apparatus for accuracy. R, repeat, so we get reliable data. Boleh? Okay. The first one, to elaborate R, okay, we repeat our experiment, so we will get reliable data. So how to make the experiment data reliable? Repeat the experiment. Another way is to increase number of sample. Contohnya macam dalam satu eksperimen, kita guna quadrat sampling technique, let's say. You guna 3 je quadrat spot untuk dapatkan data for the quadrat sampling technique. Adakah cukup untuk kita dapat figure for the population size? Boleh. Tapi kalau kita nak lebih bagus, boleh reliable data, repeat ataupun increase number of quadrat yang kita nak buat. Repeat the number of spot, tempat yang kita nak letak kita punya quadrat dan kita cari bacaan dia. Okay, so kalau kita increase number of sample, lagi akan menyebabkan kita punya data tu jadi reliable. Okay, so that kita boleh identify anomalies, lepas tu cancel dia. Kalau anomalies ni kita tak boleh pakai nilai tu sebab dia out of our range. So kita terus buang, tak boleh gunakan dia dalam kita punya calculation for average and the next step dalam eksperimen tu. Okay. Example. Okay, please refer to question on page 4. Okay, on page 4. Jumpa tak? Okay, handouts on page 4. Suggest one way of improving the method used in this investigation. Investigation yang mana guys ni? Ini adalah eksperimen yang EGA tadi kan? Cuba tengok balik. Dia kata suggest one way to improve. Maksudnya dalam eksperimen dia tadi ada something yang dia mungkin boleh menjadi source of error. So how to improve? Alright. Dia tak ada pun repeat eksperimen. Maksudnya tadi dia buat blok agar blok A, B, C, D seketul dia setiap satu. Lepas tu dia check the time taken for the EGA turn from green to red kan. Sekali je dia baca. Mana dia tahu tu betul ke tak? Mana tahu kalau dia buat 3 kali dia dapat 3 bacaan. Yang pertama tu tak okey. Macam teacher bagi tadi kan 5, 15 dengan 17. Kita kena cancel yang pertama sebab pertama tu 5 minit je. Berbeza dengan the second and the third. Okay, so it is very important to improve our investigation by repeat the investigation at least 3 kali. Okay, boleh? Okay, next question kat bawah dia ada sambungan satu question ni. Ni bukan dalam describe repeat experiment tau. Okay, yang ni dia suruh describe safety precaution. Dalam source of error, safety precaution ni bukanlah source of error. Cuma I nak sambung terus je dengan question tersebut. Okay, describe how step 2 could be carried out safely. So dalam step 2 dia kata, the student use a ruler and a knife to measure and cut 4 blocks of agar, A, B, C and D from a larger pieces of agar. Each block was a different size as shown in figure 1.1. Yang tadi tu block A, B, C, D kan. So macam mana cara kita nak tulis safety precaution kat sini? Nak cut them guna knife and ruler. How to write supaya macam nak tulis, nak handle this activity dengan selamat. So whenever you use knife and you want to cut something, you tak boleh cut on the air like this. You kena cut onto a surface. Okay, this is the answer. Cut away from the body, ataupun maksudnya janganlah potong macam ni, potong ke luar, ke arah luar. Away from body, or I suka jawapan, tak nampak lagi. Cut on flat surface. Nak potong bawang, tolong your mum masak, nak potong bawang kan potong atas surface kan, atas chopping board tu. Jangan potong macam tu. Bahaya. Potong atas chopping board or cut on a flat surface. Okay, boleh. That is our safety precaution. Yang ni nak guna pisau kan, bukannya nak panas ke apa ke. So kalau panas kita guna glove. This one nak cut, so kena guna flat surface. Okay. Next source of error, kegagalan memilih apparatus yang betul, okay, will cause us to get an accurate data. So how to get accurate result? Use suitable apparatus. Example dia, okay, use burette instead of measuring cylinder. Burette lebih accurate compared to measuring cylinder. To maintain temperature, use water bath. Boleh. Use weighing scale ataupun electronic balance untuk dapatkan mass. Okay. Electronic balance lebih accurate berbanding weighing scale yang biasa kan. Yang pusing ataupun kalau yang digital tu pun not bad, accurate. Tapi the best is electronic balance lah. Electronic balance ni bukan ke yang ada apa tu? Cover by the trimming kan, so dia lebih accurate. Okay, tapi depends on experiment, bukan semua kena pakai balance yang too accurate. Kalau kita just nak ukur mass of potatoes, kita boleh guna electronic balance yang ada kat lab tu. Alright, next example of this question untuk jawab accuracy tu, turn to page 16. This question on page 16 regarding the balloon study tu. Balloon and string were used to collect and measure the gas produced by the yeast cell. Suggest another more accurate method of collecting and measuring the gas. Dia dah mention accurate. So nak bagi dia lebih bagus, to improve, kita kena guna apa? Accurate for apparatus yang betul. So bila nak bagi, suggest another more accurate method. Use another, change the apparatus yang kita nak pakai. Tak payahlah ukur the circumference, tapi kita boleh ukur volume of the gas produced. Macam mana nak ukur volume of the gas produced? Kita guna use gas shrink. Okay, kalau guna gas shrink kan kita tahu they have scale. Directly kita boleh read the volume of gas produced. Tak perlu nak ambil circumference. Kalau nak ambil circumference, sometimes the balloon itself ada dia punya different thickness. Bila different thickness, so dia mengembang pun tak sama. So dia dah boleh kacau kita punya result. So result kita jadi tak accurate. So that's why lah, it is very important to choose correct apparatus. Okay? Boleh kita go to the next question. Okay, V for variable. Variable kena sama. Constant variable, dia kena constant, kena sama. Barulah kita boleh dapat valid result. Okay, how to get valid result? Fix the constant variable. Always use same. Cut the film into 1 centimeter long for each experiment. Same. Okay, use same type of protease. Use same source of protease. Macam eksperimen kita buat tadi tu kan, yang tu film tadi tu kan. Alright. So use word same. Same ni pun kita boleh letak dekat kita punya planning experiment so that kita boleh dapat markah CV. Right. This is an example, but this one is June 2020. Variant 6.1. Tak ada dalam handout korang tu, tapi kita baca sama-sama. Kita tengok soalan ni dan kita cuba jawab sama-sama, okay? Alright. Scientists investigate the effect of different concentration of sodium chloride solution on the germination and growth of marram grass. Kita jumpa apa? Different concentration of sodium chloride solution adalah the MV. And germination and growth of marram grass is the RV. Marram grass seed were germinated in petri dishes on filter paper which had been soaked in one of the different sodium chloride solution. Each petri dish contained 15 seeds. Perkataan. Each petri dish contained 15 seed each. So terus jadi CV. Constant variable. And the investigation was repeated for 4 times. What is this? This is to get reliable data. Reliable data. After 20 days, the length of the seedling roots were measured with a ruler. Length of the seedling root were measured. This is how they get the RV. How we get the RV. And then the question. State the variable that was changed. Independent variable and the variable that was measured. So what is the independent variable? Dah, kita dah tulis dah. So ambil je lah from yang atas ni. Tulis kat sini. Then, betul? Different concentration of sodium chloride solution. Dependent variable dia apa? Germination and growth of marram grass. Kalau kita refer this one pun also can, boleh je tulis length of the seedling root measured after 20 days. Boleh? Okay. Next question. Yang ni. Ni question yang terkini tau tahun ni. June series baru-baru ni. State 2 ways the scientists designed the investigation to produce valid result and reliable result. Nasib baik kita dah belajar tadi. Nak dapat valid result kena apa? Kena buat apa? Variable kena fix. Kena sama. Okay. Reliable nak buat apa? Kena repeat. Jumpakah kita this dua perkara ni daripada apa yang saintis tu buat? Jumpalah. Okay, so fixed variable. Each petri dish contain 15 seed. As I mentioned, bila dia kata each, it's like same number. Each of the petri dish will be put 15 seed. Okay, seems like it is a constant variable. So, bila dia constantkan variable tu, the number of seed is the same for each of the petri dish, automatically kita akan dapat valid result. Sebab kita fix the variable. Okay tak? Clear tak everyone? MRSM kucing okay? MRSM batu pahat okay? MRSM TMT ni Tun Stephen, is it? TMT ke TMS? Ke Tun type? Sorry.
Tun Mustafa Tawau.
Yeah, okay. Tun Mustafa Tawau. Alright. Jauhnya. TMT okay? Tun Mustafa Tawau okay tak? Tahu okay? Boleh dapat tak this one? Be valid for?
It can be Penang Mutant Ninja Turtle.
Okay. Alright. Do you get from the source of error yang kita understand tadi, kita boleh jawab this question. Okay, this is the latest way how Cambridge asked us. Okay, so far I tak jumpa lagi in previous years from 2018 until 2020. Okay, 22 ditanya like this. So I hope that when you understand what is VAR, you can give answers for this easy peasy. Boleh everyone? So, this is the answer. Just ambil je daripada ayat dekat atas tu. Pada question tu sendiri ada jawapan dia. Cuma kita kena understand which one is the answer for this, which one is the answer for that. Okay, so untuk valid, okay, number of seat is fixed into 15 seat for each petri dishes. Alright. Untuk reliable, the investigation was repeated 4 times. Clear? Senang kan? Okay, I hope that kalau keluarlah soalan November series, semua orang easy peasy. Memang 2 markah tu dalam tangan. Benda yang senang kita boleh jawab kan. So, by understanding of VAR, kita boleh handle all the question easy. Okay, so jangan lupa pasal VAR. Boleh? Alright, I hope that everyone boleh faham that part. Okay, next. Ada 2 lagi, ada 3 lagi command words yang kita nak discuss. Okay, kita terus pergi kepada drawing and magnification. Yang ni senang. Okay, yang ni senang, semua orang boleh buat. So, bila kita nak buat drawing in IGCSE, as stated previous pada awal tadi, you must use HB pencil. HB pencil ni dia punya LED tak terlalu terang, so that nanti bukan tak terlalu terang, dia tak smudge bila you lukis. Okay, memang Cambridge pesan, okay, use HB pencil. Your drawing must be clear, must be sharp, unbroken line, continuous outline. Detail drawing kena ada, okay. No shading, tak payah color. Okay, label line should be drawn with ruler. Kalau dia suruh label, use pencil together with ruler. Tak boleh main garis freehand macam tu je. Kena guna pencil dengan ruler untuk labeling. Okay, example. Turn to page 18 of your handouts. Okay, the question asks you to draw a large diagram of the flower in figure 2.1. Okay, untuk drawing, first tip. Okay, trace on the diagram. Masa you trace, lu tenung betul-betul gambar tu and then trace guna pencil atas diagram tersebut. Identify all the details of the gambar. Selagi you boleh trace, selagi tu you cuba untuk trace. It means that selagi you boleh trace, itulah yang you kena lukis. Okay, so masa kita tracing tu, kita dah baca, oh kat sini ada lekuk-lekuk, kat sini I boleh tak I trace inside the diagram tu? Yes, boleh. Selagi you boleh trace, selagi tu you kena lukis. Okay, dalam case ni, I dah trace dah tu. Okay, dia punya marking scheme. The MP1, marking point number 1, biasalah single clear lines, okay, for outline. Use at least half available space, okay. And then the detail for this diagram, at least 6 sections indicate with lines on either side. Dua side ada at least 6 section. And the detail number 2, at least 3 overlapping section or indication of zigzag line down the center. Okay, I bagi satu contoh jawapan ni. You rasa this one dapat berapa markah? Outline. Kalau I, for this diagram, outline dapat 1 markah because the outline very clear, there is no unbroken line. Okay, use at least half available space. So because this one, the picture I dah potong, I dah crop. So kita tak nampak how the portion gambar ni with the ruang yang diberi. But I believe this is big enough. So for size, I bagi 1 markah. And detail, at least 6 section, each line, each side. So ada 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So dapatlah detail 1, 1 markah. Detail number 2, at least 3 overlapping section or indication of zigzag line down the center. So ada this one, 1, ada overlapping like this. So dapatlah detail number 2 dia, 1 markah. Total 4 marks for this diagram. Okay? Another examples. This one. Outline. Berapa markah for outline? I would say 0. Why 0? Because there is smudge over here. Ada sikit-sikit macam broken line over there. So outline dapat kosong. Alright. Size, I give size 1 markah. Detail number 1, dia ada 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Tapi ni bolehlah kira. So bagi 1 markah untuk detail number 1. And detail number 2 ada overlapping here. Overlapping like this. So 1 markah. Total 3 marks for this diagram. Setuju? Okay. Next. How about this one? Outline 1 mark. Size bagilah 1 mark. Detail number 1. 1, 2, 3, 4, 5, 6. Detail number 2. Tak ada overlapping kat tengah ni. So dapat kosong. Total 3. Boleh? I hope that everyone's still okay. Okay. Next question. On the same question paper, but this one is on page 6. If I'm not mistaken, check on page 6. Betul tak soalan ni? Ke I and then the details, proportion and the filament and the lamella shown, there is 14 to 8 branches, at least one alteration of branches. So daripada 4 pilihan, proportion nombor 1, this one number 1, ada filament and lamella shown, ada 14 branches or 14 to 8 branches, and then at least ini number 4. One alteration of the branches. Kita tengok this one. So berapa markah this one? Outline. Yeah, outline 1 mark. Very clear. Larger than the size. Yes, I think this one is larger. Tak nampak portion yang sebenar sebab I dah crop. MP3 and MP4 portion filament dengan lamella tu portion bernice. Okay, detail number 1 saya bagi 1 markah. Detail number 2, ada lagi tak? Portion okay, filament and lamella shown. Yes, this is the filament, this is the lamella. So lamella and filament ada, so 1 markah. Terus dapat 4 marks. Alright, lagi. Yang ni berapa markah? Outline. Nampak tak? Bila kita guna pensel yang tak sesuai, bila you padam, dia ada macam conteng-conteng. So yang tu dah hilang markah nanti untuk outline. Can we use mechanical pencil? I tak advise you guna mechanical pencil. But if the mechanical pencil using lead, HB lead, I think boleh. Okay, right. But boleh kalau you dah biasa guna mechanical pencil, asalkan dia HB, okay je, don't worry, okay. Alright, cuma mechanical pencil ni takut tekan, dia tekan sangat nanti dia jadi dia left texture, okay, texture yang nanti jadi tak I risau gambar tu jadi tak kemas. Bila tak kemas, ada shading nanti hilang markah untuk outline. So be careful if you use the mechanical pencil, okay? Alright. Next, size. So this one I bagi kosong. The size is okay. The detail number 1, portion okay. And then detail number 2 ada lamella, ada filament. Ada filament, ada lamella. Terus dapat 1 markah. Nampak macam susah tapi senang je nak score marks. You kena cuba. Dah cuba baru nampak. Okay. Okay. Alright guys. Tak keluar lagi. Alright, calculation and magnification. Seterusnya kita punya common word calculation dengan magnification. Lepas drawing, kenalah calculate and magnify. Kita punya gambar rajah tadi tu. Okay. Untuk calculation and magnification, show your working. Show your calculation. Give your answer to the correct number of significant figure or decimal places stated in the question. Question biasanya dia akan trick kita kat sini. Dia nak berapa significant figures, dia nak berapa decimal places. Korang kena baca question dengan betul dan underline supaya you tak miss, okay, point-point yang dia minta tu. And then give the correct units. For magnification, cara nak tulis magnification, times is at the front. Then come the number. And any measurement must be in mm. Wajib bila kita melibatkan magnification. This is how you write down and this is the unit. Lain daripada unit ni, nanti korang salah. Okay, guna unit yang tepat. Okay, refer to the page 6 of your question paper now. Okay, sekali lagi question ni akan minta you measure. Betul kan? Measure the length of line PQ in figure 1.3. So, have you measure? Apakah panjang PQ? Kalau you, if teachers print bahan tu actual size, that the result would be 90 mm plus minus 1. 90 plus minus 1 mm untuk PQ, panjang PQ. Okay, and then give your answer. Dia suruh kita calculate the actual length. Okay, calculate actual length of the part of the fish gills using the formula and your measurement. Give your answer to 3 decimal place. Markah dia 3. Satu markah kat sini. And then satu markah bila you fill in the formula. And another one mark if you ikut dia nak apa. 3 decimal place. So cara dia senang je kan. Actual length sama dengan 90 daripada sini. 90 divide by magnification. Magnification ada pada gambar. Times 550 kan, okay, and then you will get 0.164 something. Tapi the question asks you 3 decimal place, so stop sampai 3 decimal place sahaja. So dapat full mark 3. So ni memang senang, okay, ni memang senang.
Teacher.
Yes.
There is a question.
Right.
If we show that gambar as a portrait, can we draw like a horizontal?
No problem. Can. Boleh je. Okay. Let's say macam gambar tadi dia suruh kita lukis like this kan.
Yeah, thank you.
Dah macam Rian. Tiba-tiba you draw dia sebab you nak buat besar kan, so you buat macam ni. Boleh.
Yes.
Boleh. No problem.
Yeah.
Okay.
Oh, so we can draw it like a horizontal one lah. Kan?
Yes.
Betul tak?
Yes, can.
Thank you, teacher.
Right. Okay, guys. So, can we proceed to this question? Turn to page 7 of your handouts. Okay, measure the length of the line PQ, guys. So, I dapat kat situ, if it is the exact papers yang tak tukar dia punya size, memang size yang exact yang cikgu print, so you will get this measurement, 62 mm plus minus 1 mm. Okay, and then calculate the actual length. Again, they ask for actual length of the pollen grain using the formula, this one, and 2 decimal places. Okay, so this is our answer. Boleh ya? Easy kan? Boleh? 2 decimal place, so 0.4 mm jawapan dia. Right? Alright. Next question, this one is calculation. So far for 2021 dengan 2022 ni, I nampak selalu mesti tak miss question asking for percentage decrease. Okay, so for this question, page 21, scientists investigated the effect of wind speed on the average distance traveled by single pollen grain and group of 5 pollen grain. So ada dua kriteria, satu single, satu lagi group. The pollen grain were dropped from a height of 2 meter. And then the result of the investigation are shown in table 2.1. Dia dapat satu travel single, satu lagi travel clump. Okay. The question is, calculate the percentage decrease in the average distance travel when the pollen grain fell as a clump compared to single pollen grain at a wind speed of 0.6 minutes. So, kita punya data, kita nak tengok yang ni je. So, ini sahaja fokus kita. Dia kata average distance decrease. So, kalau decrease, inilah cara dia. Right? So, 55 minus 118 divide by 55. Mana yang rendah? Sebab dia nak decrease kan? Rendah, turun. Okay, 50 lah kat bawah, 55 kat bawah times 100. So you will get 53. 55 minus 118, of course saya dapat nilai yang negative. But in IGCSE, they give you, dia in bracket kan negative. If you put negative, okay, tak pun tak apa. But as you know, 55 minus 118, of course saya dapat nilai negative. So, maintain a negative reading. Okay. Because of the question are percentage, so letaklah dia punya unit as a percent. Alright. So, next. Last techniques hari ni, that is how to draw graph. Okay. Alright, for graph. There is so far latest for 2021 and 2022, I saw the question always asked about line graph, ask you to draw line graph, histogram and also bar graph. Okay, hari ni kita nak tengok line graph dengan histogram. Okay, bar graph I believe semua orang pun understand macam mana nak lukis kan, tak susah. Line graph ni dia ada beberapa case, okay. Alright, line graph make use of the space given. Biasanya kita dapat one set of paper graph tapi satu bahagian tu je kan. So fully utilize that space. Okay, IG dia memang bagi that space je. Dia tak bagi one paper, graph paper macam tu. Because dia nak yang tu je. Size tu dah determine and then dia mesti, your graph must be 3/4, cover the 3/4 of the space given of the graph paper tu. Okay, when you want to plot, you can use cross or dot in a circle. You boleh buat dot. Katalah this is our kertas graph. You boleh buat dot and then circlekan the dot. That is what meant by dot in a circle. Okay, must join all dots using ruler. This is called as line of best fit. In biology, line of best fit, you just join the dots using ruler and pencil. That is line of best fit in biology. No extrapolation. Okay, if there is two sets of data, label each line graph or you boleh write key. Okay, lately the question asks you, bila dia bagi dua set of data, you kena buat key for the graph. Okay, macam mana ya? Mari kita tengok contoh. So on page 10 of your handouts, dia bagi this. Right. Dia bagi the vitamin C concentration of each cabbage leaf sample was then determined. The result are shown like this. So they ask you to plot a line graph on the grid of the data in table 2.1. So dia dah bagi, sorry, hang sikit. Okay. So dia dah bagi, memang this je the graph. So there is space over here for you to write all your y-axis and your scale. And there is space here, enough space for you to put your scale over here and also label your x-axis. So bila kita nak jawab graph, jumpa je space to jawab graph. We terus tulis dekat sini. Sorry. This is RV. This is MV. You kan, when we get question, kita akan go through, right? Kalau dah boleh tulis, terus label RV dengan MV, so that nanti you tak dah kalut sebab ada 60 minit je nak jawab semua sekali soalan kan. So make sure that you grab this marks, okay? Jangan kalau tu tak tahu nak tak yang mana. So daripada kita punya table tadi, kita dah tahu this is our MV, this is our RV. So terus translate it into data kita kat sebelah ni. RV dia apa, MV dia apa. So write down your title of your X and title of the Y. Okay, and I advise you bila nak label, bila nak start to do the scaling, only use this one. Yang you nampak ni je. Okay, bukan I tak suka, but I believe that Cambridge, they have already set a set of graph. When they dapat your answers, they just put that graph onto your graph. So the scale will be using this. That is reason why Cambridge give you the 10 blocks, this one, okay, and the smaller one. So your scale should be in at here, at here, at here, and here. Okay, I don't like if student the scale will be too crowded, not nice. Okay, so untuk this one, if you sempat buat, this is the answer. Boleh? So we only sambung, okay, plot the lines is only from point to point. Jangan lebih-lebih. Kalau if that is extra like that, sikit je, tapi dia melebihi daripada four small blocks tu, this will consider as extrapolation. Extrapolation, you akan lost mark for line. Kalau kita nak lukis graph, kita ada axis, okay, axis satu markah, and then scale satu markah, and then plot. Duduk ni tulisan teacher. Plot 1 marks, and then last one is line 1 marks. Okay, so if there is extrapolation, you akan hilang this mark. Okay, so do not extrapolate atau memandai-mandai sambung line. Kalau tak ada data, jangan pandai-pandai extrakan your line. So just sambung point to point by using pencil and ruler. Okay. Next question, and this one lately memang ada question like this. Okay, use your graph to estimate the concentration of vitamin C in a sample of cabbage leaf that has been boiled for 20 minutes. Show on your graph how you obtain your estimation. So pada your graph, identify dekat 20 minutes. Okay, boleh nampak tak graph I tu warna kuning tu? So sambung dia sampai touch your graph and then go to the y-axis. Then you will get the result. So whatever you get from your graph akan dapat satu markah. Transfer this data to the space given, that is 17.5. So the 2 marks will be given here, 1 marks, and also for the line 1 marks. Setuju? Boleh ya? Okay guys, next question on page 21. This case, there is a wind speed. Yang case pollen grain tadi ada wind speed. Dia bagi average travel by single pollen and average travel by the clump pollens. 2 data. And then they ask us to plot a line graph on the grid of the data and include both set of data and key. So refer to the key, meaning that kita kena pilih kita punya this one you nak guna plotting macam mana, X ke, this one macam mana, mungkin point macam tu. Lepas tu you tulis the key dekat sebelah this area. Boleh. Again, what is on Y? Y mestilah RV. What is on X? X mestilah MV. MV kita apa? Wind speed. This is MV. This is both RV. Kan? Senang kan? Senang sangat. This is the result. Comel kan? Comel kan? Okay. So, untuk data tadi, kalau you perasan, data kita paling tinggi sekali 154. Okay. So, pada your graph, this is wajib. Walaupun 154 and last ke ujung tu, kita boleh label 160. Label. Jangan tinggal. If you lupa nak write the 160, nanti you hilang marks for skill. Okay, so whatever your data, your skill mesti higher than the data. Boleh? Okay, so this is what I mean by key. Okay, kalau you nampak, here I use bulat macam tu. Cuma dekat sini I should tambah. Okay, like that. Why dia minta kita kalau kita nak tanda macam ni and then kita circlekan? Because apa-apa dalam circle tu sahaja yang kita libatkan. Tak ada extrapolation. Nak selamatkan you. Alright. Now, clear? If there is two graph, two sets of data need to be plotted onto your graph, please make a key like this. Okay, next question. Again, use your graph to estimate the average distance traveled by single pollen grain at a wind speed of 0.5 meter per second. Show on your graph how you obtain your estimate. Ta-da! So, dekat mana? Dekat 0.5. This one will get 1 mark sebab you draw the line. This one transfer into here, 94. You will get another 1 marks. Okay, 2 marks here. Clear? Okay, next on page 25. Okay, a student monitor their pulse rate after exercise. The student pulse rate before exercise was 62 beats per minute. The result are shown in table 2.1. Plot a line graph on the grid or the data and include a line of best fit. Jangan risau. Jawapan dia simple je. Because best fit line dalam biology hanyalah you sambung point to point guna pembaris. Simple je. Okay. Jangan risau. That's it. Alright. Last one. Page 26. This is histogram. Okay. They give you a population of one species of Nautilus. Okay, was studied. The width of the Nautilus shell were measured and recorded. The results are shown in table 2.1. Plot a histogram on the grid of the data in table 2.1. So dia suruh, dia dah cakap siap-siap. Histogram. So dia bagi kita selang kelas like this. Okay. 1.101 until 110 ada 8 shell. So macam mana nak buat histogram? So this is how our histogram look like. Sama juga. This is RV. This is MV. Both must have unit. Kalau reading kita tu ada unit. Number of shell tak ada unit. So tak perlu tulis unit. Okay. I hope that everyone, yelah kalau we have plenty of time, maybe you all boleh jawab dulu baru tengok jawapan ni. So I hope that teachers at maktab ask the student to hand in the handout so that kita boleh check whether the student able to draw or not. Okay. So dalam case ni, again kita ada di reading tadi, highest kat sini bukan kita punya label mestilah lebih daripada highest data yang kita ada dalam the table. Okay. Can? So I believe that this is my last explanation about the histogram. Histogram, as you understand, mesti touch bar towards bar to another bar. Okay, kalau bar graph barulah dia ada space between each of the bar. So histogram sambung like this, touch one bar to another bar. Okay, boleh? Alright guys, so I believe this is last one of my slides. Okay, so I hope that everyone can score paper 6 as easy as kita share tadi. Okay, I hope that in mock IGCSE later you can apply this, okay, all the techniques that I've shared just now, okay, and boleh score 40 marks. Like seriously, paper 6 ni bukanlah paper yang susah. Every year pun memang grade threshold dia tak akan rendah daripada 30. So dan kita memang boleh dapat pun 40 marks tu. Okay, but you need to practice. You need to practice, terutama sekali for planning experiment. Memang dia ada 6 markah dan dia bukan susah pun nak jawab. Okay, I dah bagi tips-tips tadi kan. Kalau you cakap you repeat the experiment pun you dah boleh dapat markah. So jangan biarkan line yang banyak tu kosong. Dia nampak macam banyak, macam menakutkan 6 markah tapi sebenarnya tak susah pun. Okay, kita kena cuba. Baru kita tahu kita boleh dapat tak. Jangan biarkan question tu kosong. Sebab I biasa nampak pula my student also biarkan kosong je. Jawab yang lain. Padahal kalau you letak 2-3 jawapan based on yang kita share tadi tu, dah boleh dapat dah 3 markah at least. So, jangan tinggal, you kena try. You kena try baru you tahu. Okay, so that's it from me. Whatever I share today benefits you. So good luck for your IGCSE and November Series 2021. Okay, bye everyone. Thank you. Assalamualaikum. If there is any question, boleh tanya sekarang. Mungkin boleh jawab terus. Ada question?
Boleh join strategy kita.
Okay, thank you so much, Cikgu Ida, Teacher Ida. Thank you so much for the presentation, for the nice presentation. And we wish and we doa to all students will get A star for biology IGCSE. Inshallah, Amin.
Inshallah, Amin. Okay.